Good afternoon, class, and welcome to Mr. Brandis' PowerPoint presentation on evolution. This is the first PowerPoint presentation out of 5 that I will be posting. In this PowerPoint presentation, I'm going to talk to you about some misconceptions that you may have about evolution. I'm also going to talk about allele frequencies, and I'm also going to talk about Hardy-Weinberg equilibrium. So we'll get started. First off, when you look at this picture of gaming consoles slash, controllers, what I want you to notice is the more buttons you get, the more advanced the controller seems to become. In addition, you could also see that there's multiple systems shown here. Not only do we have the Wii, we have the Xbox, we have PlayStation, the three biggies. As you get more advanced, you're able to do a variety of different things. So if we go all the way back to something like the Tandy, all you were really able to play with one stick and one button was essentially Pong, which was kind of boring. And with the Wii remote, you could play things like Zelda. You have a lot of different movements, ability to be in a 3D environment. So pretty cool things. And notice that these things changed over a long period of time. The oldest one that I can actually remember is the Atari and the NES. And I recently got a Wii, so we'll see how that works out. Regardless, moving on to evolution, a lot of students think that an individual can evolve, and that's not necessarily the case at all. Only a population can evolve, especially when we're talking about generations. One person cannot evolve. It takes a group of individuals that comprise a gene pool. So only populations can evolve. The other thing is that a lot of people feel as though, oh, well, this animal had to survive. That's not necessarily the case. Evolution is not goal-oriented or directional, which basically means that it's a reaction to something. Things happen not necessarily for a reason, they just kind of happen haphazardly. So no planning or anything else like that. These things just occur and, cause and effect, right? Something happens and things change or they don't. Speaking of which, evolution is not need-based, which means that things will survive if they're good enough. There's a lot of imperfections in this world, including our own mitochondria. We only use a fraction of the energy that we could get from sugar. That shows a very big inefficiency, but apparently that inefficiency was good enough to outlast all of our other competitors. So again, evolution is not need-based, it's not goal-oriented or directional, and individuals cannot evolve. Now, if there's any other misconceptions, please let me know, write them down now, and I'll be able to talk about those in class if you feel as though you have one. So, evolution. Evolution is defined as a change in the allele frequency of a population over generations. So, first question is, what's an allele frequency? Well, the allele frequency is the number of copies of a particular allele divided by the number of copies of all of the alleles. This will make more sense in just a little while, but what I want you to think of evolution as is a biological history of our entire world. So, back to the allele frequencies. What is an allele? An allele is an alternative form of a gene, and it's one member of a pair that is located at a specific position on a specific chromosome. Don't forget, we're talking about homologous chromosomes here, and you get those from your parents, one from your mom and one from your dad. And depending on what form of gene your mom or dad gave to you, determines what you will have for a genotype, whether or not you're a homozygous dominant or homozygous recessive or a heterozygote, will determine what characteristics you have. So here we see locus for flower color. So these are the alleles, which are the alternative forms of those genes. One for white and then one for purple. So with that, we can move on to allele frequencies. Now don't forget, in the back of your mind, I want you to think that it is the number of copies of a particular allele, be it dominant or recessive, divided by the total number of copies of all the alleles. So let's imagine that we're in a field of flowers and we have 500 flowers. Now, out of that 500 flowers, we have 320 individuals that are homozygous dominant. 160 of those flowers that are heterozygotes, and we have 20 individuals which are homozygous recessive. Granted, you would have had to have done some sort of genetic study on the red flowers because both heterozygote and homozygous dominant will get you that red flower type. So you need some sort of technology in order to figure this out. But if you have it, fantastic. So assume that we have the technology and we figured all this out. So what we need to get is the number of a particular allele, in this case big R or little r, divided by the total amount. So the total amount of the alleles out of 500 flowers would be 1,000. Because don't forget, each of these flowers, each member of the 500, has 2 alleles per flower. One given from mom and one given from dad. In order to find our particular allele, let's find little r first. All we need to do to find little r is multiply 20 by 2. This will give us 40. And we have 40 little r alleles. Because don't forget, out of 20 plants, there are 2 alleles for a plant. So 40 little r alleles. We can do the same for the homozygous dominant individuals. So 320, each of them containing 2 big R's. So we multiply 320 by 2 to get 640. So the problem comes with the heterozygotes, because you have to keep in mind that you don't need to multiply anything, and because each flower has one of each allele, all you have to do is just take that number and add it to your other big R or little r numbers. We can take these plants and say, okay, we have 160 little r's and 160 big R's. So what we do in order to find our particular allele is we take our 40 that we got earlier and we add it to 160. This will give us 200 alleles, so that means we have 200 little r alleles out of 500 plants. But don't forget, each of those plants has 2 alleles, so we need to divide it by the total number of alleles. So we take 200 divided by 1,000, and we get 0.2 little r's. This is the allele frequency of little r. Here we have the allele frequency of big R, and we did the same thing that we did over here to the big R's, because don't forget, 160 plants have 160 little r's and 160 big R's, because it's a heterozygote. So we take our original number of 640 big R's that we got from these individuals, and we add it with 160, giving us 800. We take that total 800 alleles, big R alleles, and we divide that by 1,000 to get 0.8 big R alleles. Now, when we're worried about the Hardy-Weinberg equilibrium, we'll represent the dominant allele as P and the recessive allele as Q. This is just to alleviate any other confusion so you know you're not talking about other alleles. So the allele frequency of P will be 0.8 and the allele frequency of Q will be 0.2. Don't forget, evolution is a change in these allele frequencies within a population over generations. This can actually be used to test whether a population is evolving or not. Sexual reproduction alone does not lead to evolutionary change within a population. And this is because although alleles are shuffled, the frequency of these alleles and genotypes in the population does not actually change. Similar to shuffling a deck of cards, you'll deal out different cards, but the cards and suits in the deck still do not change. So the Hardy-Weinberg principle states that allele and genotype frequencies within a sexually reproducing diploid, means a full set of genetic information, diploid population will remain in equilibrium unless outside forces act to change those frequencies. So that's the general idea behind the Hardy-Weinberg equilibrium, is to basically say, are we evolving? Nope, nothing's changed, we're all set. So that's the whole point of this equation. Just to give you an example of the Hardy-Weinberg equilibrium, we'll first take a look at an animal. In this case, I'm going to be using the blue-footed booby, which is found in a couple of remote places around the world. And blue-footed boobies are interesting creatures. They are birds, and they can either have webbed feet or non-webbed feet. Here's an example here of webbing, and this is non-webbed. So let's assume that big W is a dominant allele for a non-webbed blue-footed booby. And the recessive allele, little w, is the allele for webbed blue-footed booby feet. This individual right here could be little w, and that's it. This individual, however, could be big W, homozygous dominant, or big W, a heterozygote. So let's do the same numbers just to keep everything nice and simple, and let's consider the gene pool of a population of 500 blue-footed boobies. 320 are homozygous dominant, just like the flowers. 160 are heterozygotes. 20 are homozygous recessive individuals. So the only ones that are actually going to have webbed feet are 20 out of the 500 blue-footed boobies. We can take a look and get their allele frequencies by doing the same process and adding these together and then dividing by 1,000 to get our allele frequencies. So our allele frequency of the dominant allele is 0.8, which gives us 80%. The frequency of the recessive allele was 20%. Don't forget, when we're talking about dominant or recessive, because we're going to be plugging this into the Hardy-Weinberg equilibrium, we have to use P for the dominant allele and Q for the recessive allele. So this right here is the Hardy-Weinberg equilibrium, and this is the frequency of all three genotypes that are represented within this equation. P squared represents the homozygous dominant individuals, while 2pq represents the heterozygous individuals, and then q squared represents the homozygous recessive individuals. Don't forget, all of this should add up with 100%. Don't forget, this equation should work out to 100% or 1. Otherwise, we'll know that evolution is actually taking place. So what about the next generation of blue-footed boobies? The probability that a blue-footed booby sperm or egg cell will carry the big W is 80%. And the probability of the sperm or egg that will carry a little w is 20%. So we can use these to figure out our genotype frequencies, and we can set it up like a regular Punnett square in that we have heterozygotes mating with other heterozygotes, and all you do is you multiply them together. So 0.2 times 0.2 will give you 0.4, and 0.2 times 0.8 will give you 0.16, and 0.8 times 0.8 will get you 0.64. P squared, 0.64, PQ is 0.16 for our heterozygotes, and Q squared is 0.04. These are our genotype frequencies, which are 0.64 big W, big Ws, 0.32 heterozygotes, and 0.04 homozygous recessive individuals. And again, these are our allele frequencies, which we got earlier. Don't forget, a population within Hardy-Weinberg equilibrium is not undergoing any sort of evolution. It's staying stagnant, which means stays the same. Alleles and genotype frequencies will not change unless something acts on them to change the gene pool. For a population to remain in Hardy-Weinberg equilibrium, it has to exemplify 5 different traits. 1, it must be a very large population. 2, no gene flow between populations, which means no one mating with anyone else or anyone else coming into that population and mating with other individuals. No mutations. Random mating, which means that you're shuffling that genetic deck often, as we talked about with the cards beforehand. And finally, no natural selection. Essentially, nothing stays in Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium could also be used by public health scientists to figure out how many alleles could be in a given population. So let's take Caucasian newborns with cystic fibrosis. In the United States, 1 out of every 3,300 individuals have cystic fibrosis. So the disease, which causes digestive and respiratory problems, is caused by a recessive allele. So you must have 2 recessive alleles in order to have that disease. With that statistic, we can take a look and we can say, okay, well, if Q squared represents the number of recessive alleles within that population, we can take 1 divided by 3,300 and get 0.0003. The frequency of the recessive allele is Q, which equals 0.0174, that's Q squared, or 1.7%. Don't forget, the other alleles have to be taken into account. So if you don't have recessive alleles, you have to have dominant alleles. So if we take 1 minus 0.017, we get 0.983. So taking a look at the heterozygous individuals within that population, which is represented in the Hardy-Weinberg equilibrium as 2pq, we can do this out as 2 times 0.983 times 0.017 to give us 0.304. So that gives us around 3.4% of Caucasian Americans are actually carriers for cystic fibrosis. This generally works out to be 1 out of every 29 individuals. And so if you take a look around the classroom, next time you get a chance, if we have about 30 students, essentially one of you will actually be a carrier for cystic fibrosis. So that's pretty much it for this PowerPoint. If you have any questions, please feel free to write them down and we can talk about them in class the following day. I look forward to seeing all of your bright and shining faces tomorrow morning. Thank you very much. Have a good one.